Welcome. Today, we're going to tackle a biochemical pathway that is not only a big deal on your exams, but is also absolutely essential for your survival. We're talking about the urea cycle. We're going to break down exactly how your body deals with a constant and pretty deadly toxin.
So here's our game plan. First up, we'll get to know the villain of our story, ammonia. Then we'll introduce the hero, the urea cycle itself. After that, we'll walk through all 5 key steps of the action, see how it's all connected to other metabolic pathways, and then we'll dive into the clinical side, what happens when things go wrong. And don't worry, we'll wrap it all up with some really handy memory aids to make sure this stuff sticks for your exams.
All right, let's just start with a really basic question. Every single day, your body produces a pretty potent toxin, just as a part of its normal routine. So, how does it get rid of that toxin without, poisoning itself? Well, let's find out. Okay, first things first. Before we can really appreciate the solution, we've got to understand the problem. And that problem is ammonia. Think about it like this. Every time you eat protein, your body breaks down those amino acids for energy or to build new things. It's a process that happens constantly. But it has a downside. It unavoidably releases ammonia. And this isn't just some minor inconvenience. Ammonia is highly neurotoxic. It's especially bad for the brain. Even a tiny little spike in your blood ammonia levels can be dangerous, which is exactly why the body needs a system to get it neutralized, like right away. So, with this constant toxic threat lurking, how does the body defend itself? Well, it's time to meet the hero of this metabolic story, the urea cycle. This whole pathway, which you might also hear called the Krebs-Henseleit cycle, is our body's #1 defense against ammonia. It all goes down almost exclusively in the liver. And the goal is beautifully simple. Take 2 toxic nitrogen atoms from ammonia and package them into one single, harmless molecule of urea. And why urea? Well, it's perfect. It's chemically neutral, it dissolves in water like a dream, and our kidneys can easily filter it out into our urine. Problem solved. And what's really cool is that this cycle doesn't just happen in one place. It's a fantastic example of teamwork inside the cell. The first 2 steps, they happen inside the cell's powerhouse, the mitochondria. Then, for the grand finale, the process moves out into the main cellular fluid, the cytosol, for the last 3 steps. It's a real 2-part operation. Okay, let's get to the heart of it. We are now going to walk, step by step, through the 5 essential reactions that turn a dangerous toxin into a harmless waste product. All right, here they are, the 5 main stages of our assembly line. We're going to start by capturing that pesky ammonia. Then we'll build a few intermediate molecules, sneak in that second nitrogen we need, and then boom, in the final step, we release our finished product, urea. So let's break down each station. The whole process kicks off in the mitochondria. An enzyme called Carbamoyl phosphate synthetase I, or just CPS I for short, grabs that free ammonia and combines it with carbon dioxide. Now this is the step to remember. Why? Because it's the rate-limiting step. That means the speed of this one reaction controls the speed of the entire cycle. It's the bottleneck. And it's a pretty big commitment energy-wise, costing us 2 ATP molecules right at the start. So, next up, that carbamoyl phosphate we just made passes its carbamoyl group over to a molecule called ornithine. You can think of ornithine as a reusable shuttle bus. It just picked up its cargo, and by doing so, it forms a new molecule called citrulline. This citrulline is then promptly shipped out of the mitochondria and into the cytosol for the next leg of the journey. Okay, now we're out in the cytosol. Here, citrulline links up with an amino acid called aspartate. And this moment is critical because aspartate is donating the second nitrogen atom that we need to make our final urea molecule. This reaction creates Argininosuccinate, and it also costs us some energy. 1 ATP molecule gets broken down into AMP, which is basically like spending 2 high-energy bonds. Now this step is really fascinating because we get a split. Argininosuccinate is cleaved into 2 different things. One is arginine, which is going to continue on in the urea cycle, and the other is fumarate. And fumarate, well, that's a direct ticket into another huge metabolic pathway, the TCA cycle, also known as the Krebs cycle. We'll definitely come back to that important connection in a bit. And here we are, the final step. The enzyme Arginase comes in and just snips the arginine molecule. This releases our final product, urea. And what's left behind? Our trusty shuttle bus, ornithine. It's now empty and all set to head back into the mitochondria to pick up another load of carbamoyl phosphate and start the whole cycle over again. It's a beautiful, continuous loop. So we've just seen all 5 steps. Now let's zoom out and take a look at the bigger picture. We're talking energy costs, how the whole thing is controlled, and its crucial connections to the rest of the metabolic city. So let's talk about the energy bill for this super important detox process. When you add it all up, making just one single molecule of urea costs the cell a total of 4 high-energy phosphate bonds. That's some pricey stuff. Now, I know 4 ATP equivalents sounds expensive, but the body is incredibly clever about this. Remember that fumarate we made back in step 4? It doesn't just go to waste. It can jump right into the TCA cycle, which is our body's main engine for generating energy. This creates this brilliant connection, sometimes called the Krebs bicycle, where the 2 cycles kind of feed each other. It's a perfect example of metabolic efficiency that helps pay back some of that energy debt. A pathway this powerful can't just be running at full blast all the time. It has to be tightly controlled. And the main control knob, as we said, is that very first enzyme, CPS I. It gets turned on by an activator molecule called N-acetylglutamate, or NAG. You can think of NAG as a kind of sensor. When do we have lots of extra amino acids floating around? Right after a big protein-rich meal, right? Well, that surplus causes more NAG to be made, which then flips the on switch for CPSI, and the whole urea cycle revs up exactly when we need it most. So, this all leads us to the clinical side of things. What actually happens when this essential detox plant in our body breaks down? Well, as you can imagine, the consequences are really serious. See, you can have inherited genetic problems in any of the 5 enzymes of the cycle. If you look at this table, you can see how a specific enzyme defect gets its own name. So, for instance, a defect in Argininosuccinate synthase leads to a disorder called citrullinemia. A problem with the very last enzyme, Arginase, causes hyperargininemia. But even though each defect has its own unique biochemical fingerprint, they all lead to the exact same dangerous result. And that shared result is hyperammonemia, which just means dangerously high levels of ammonia in the blood. In the brain, all that extra ammonia actually messes with the TCA cycle, which disrupts the cell's ability to make energy. And this energy crisis is exactly what causes the neurological symptoms you see, like lethargy and poor coordination. If it's not treated quickly to get those ammonia levels down, the damage can be permanent, and sadly, it can even be fatal. Okay, we have covered a ton of ground here, so let's make sure it all sticks. We'll do a quick recap of the high-yield points, and then I'll give you a couple of tools to help you lock this in for your exams. All right, so to wrap it all up, the urea cycle is the liver's main strategy for detoxifying ammonia. It's a process that happens in 2 locations. It starts in the mitochondria and finishes up in the cytosol. It's got that clever link to the TCA cycle through fumarate, and if it fails, the enzyme defects lead to severe clinical problems because of ammonia toxicity. Okay, time for a quick pop quiz to really make this stick. Which enzyme is found in the mitochondria, serves as the main rate-limiting step, and needs N-acetylglutamate to get activated? Just take a second to think about it. Got it? If you said carbamoyl phosphate synthetase I, or CPSI, you are absolutely right. Nicely done. And finally, to help you remember the key players for your exams, here's a pretty simple mnemonic for the intermediates in order. Only cool cats are arrogant. Let's break it down. Only is for ornithine. Cool is for carbamoyl phosphate. Cats is for citrulline. R is for argininosuccinate, and arrogant is for arginine. And just like that, you've mastered one of the most important detoxification pathways in all of biochemistry. Well done.